Aloha, this is Roger Brewer with the Hawaii Department of Health. This is a recording of a presentation that I gave to the State Risk Assessors Association on July 16, 2019, entitled Fake Data, the Need for Sampling Theory in Environmental Research and Investigations. So what I want to do today is share some experience I've had over the past 25 years with data that we've collected in the environmental industry and problems we've had interpreting this data, and discuss how the problem might be more related to the way we're collecting samples than with the data itself and statistical methods that we've used. A common problem in risk assessments and with environmental data is highly variable data. For example, we might be looking at the correlation of contaminants in soil and uptake into produce, where we're comparing one concentration to another and looking for trends. Another example is vapor intrusion, where we might collect samples of soil vapors under slabs and compare that to indoor air concentrations and try to calculate an attenuation factor, predict impacts to indoor air. Another type of data we collect looks at trends. So for example, with indoor air quality, we may be collecting samples of indoor air over time, looking at the concentration and trends over time, or bacteria in swimming areas, common problem in Hawaii, where we collect samples at different times and look at concentration variability and trends over time. A lot of the data for these examples tends to be highly variable, very difficult to interpret. One of the sites where we first began our look into the sampling methods and problems with highly variable data was a 4-acre property on the Big Island of Hawaii, contaminated with arsenic, proposed to be redeveloped as a hotel site. So we collected about 15 or 20 discrete samples from the areas we normally do under a discrete sampling approach, and saw that the data were highly variable, even closely located. Samples within just 3 or 4 feet from each other maybe range from 1,000 ppm arsenic to 4 ppm arsenic. So how do you deal with this problem? When this was in 2004, we took the standard approach. You just keep collecting samples until you run out of time and money, and then you dump the data on a risk assessor. I want to mention my background is actually in geology, so I have a PhD in geology, originally in mining exploration and oil and gas exploration. I was also kind of forced to be trained as a risk assessor early on in my career. So I'm actually the person collecting the data and then the person that the data is getting dumped on at the same time. So I've caused myself, you could say, a lot of problems over the years, or definitely raised a lot of questions about what I was doing on the sampling side versus the risk assessment side. So what do you do when you have highly variable data? This example, we're looking at arsenic and contaminated soil. If you only have a few samples at a site, you might just take the maximum concentration. But this always posed a problem. What do we mean by maximum concentration? And how do I know this is the maximum concentration at the site? How does this relate to risk anyway? Where we look at means, not maximums. If we have enough sample data, we may plug it into ProUCL and let ProUCL calculate means and standard deviations and 95% UCLs and such for us. If there's a lot of variability within the data, then you'll have a lot of uncertainty in your calculated means and 95% UCLs. So how do you take care of that as a risk assessor? And it's even in EPA's risk assessment guidance. We'll just toss out the outliers. This always caused some problems on the geologist side or on the site investigation side, because I collected that sample. I know arsenic or PCBs or whatever is high in that part of the backyard or wherever I'm collecting the sample. And the idea of tossing out the outliers from a risk standpoint doesn't make sense, because we look at average exposure over time, and I know my kid is going to wander across that area randomly over time. So tossing the outliers, that was an early red flag, that something was wrong with what we were doing. Imagine this, back to the geology side, if we were exploring for gold in your backyard or gold anywhere, and some of the samples came up 100% gold, so you're actually finding gold veins and such. Would you really toss those outliers? If you did that, I can tell you for a fact in the mining industry, you'd lose your job pretty quickly. It doesn't make sense at all. But they realized this back in the late 80s when they were first writing sampling guidance for environmental contamination work. It's a quote from 1989, "All data not known to be in error should be considered valid. High concentrations are of particular concern for their potential health and environmental impact, just as they are of concern and of interest to people looking for gold in soil or in rocks." Francis Pittard, 2009 paper he wrote, actually he was saying the same thing back in the 1980s, 1990s. A common error in the environmental industry has been to reject outliers that cannot be made to fit a statistical model. The tendency has been to make the data fit a preconceived model instead of searching for a more appropriate way to collect samples. Some of the results of highly variable data that we deal with on a routine basis in the environmental industry, especially contaminated properties, we're looking at soils, the repeated need to collect additional data with no clear endpoint in sight, or manipulation of a database to misleadingly, I would say, reduce uncertainty. And ultimately, you end up with uncertainty and potential risk, uncertainty in the adequacy of proposed remedial actions, where you see failed confirmation samples following remediation. Then you end up with abandoned properties. And most abandoned properties in the U.S., especially in urban areas where land is expensive, aren't related to contamination or even to money. It's more uncertainty in the data and no clear endpoint to investigations or remediation, so the property owner just walks away from it. A bigger concern is when remediated sites are later found to still be contaminated, which is starting to happen more as people go back and test some of the sites that were cleaned up in the 80s and 1990s. So let's step back and look at what's going on here, what the potential cause of these problems are. There are actually two sides of sampling theory and two types of media that we test. The first is referred to in sampling theory as finite element media. These are elements that are individually identified and randomly selected as part of the investigations. So examples include rooms of people or a forest of trees, cans of tuna, bags of rice, bags of soil, bottles of water, where if you were going to run some test on these types of media, you would select individual elements and then run the test to answer your questions in the investigation. Well, another type of media, it's very widely known in the agricultural industry and mining industry, but essentially unknown in the environmental industry until recently. It's referred to as infinite element media. In this case, the elements cannot be individually identified or randomly selected for testing. So examples of these type of media include a pile of flour or pile of rice or pile of soil or soil in situ in your front yard or anywhere, or a lake of water, a room of air, or subslab vapors under a building. So these are all media that you have some question about, but you can't go in and collect individual grains of flour or grains of rice or bits of soil or molecules of water or air and such. So there are completely different statistical methods and sampling methods applied to each type of media. And the issue that we're seeing in the environmental industry over the last 25, 30 years, as we'll discuss during this presentation, so we've been attempting to apply sampling methods and statistical tests for finite element media to infinite element media. There's a good summary of sampling theory basic in these two documents. They're fairly basic. One is called Good Samples, Guidance on Obtaining Defensible Samples, a sample collection in the field published in 2015. A follow-up on it, Good Test Portions, called Guiding on Obtaining Defensible Test Portions or Laboratory Subsamples, just published in 2018. These were mainly written for the food industry, but really the discussions in it on finite versus infinite element media apply to any type of media that we test. So as I mentioned before, the testing of finite element media versus infinite element media are quite different. It's important to understand the differences. So for finite element media, some of the common data quality objectives or questions that we're asking in the investigation might include variability between individual elements, estimates of minimum or maximum concentrations, or other quality control parameters. It might be the weight of a bag of rice or something. So in this case, the sample is prepared by collecting and independently testing individual elements. So the only variable of importance really is the number of elements selected. So it's fairly simple. If you want to get a greater precision, better certainty in the data, you collect more elements. This is really what we learned in high school and in college, was statistical methods and sampling methods for finite element media. Now the other type of media, infinite element media, it's a lot more complex. You have to do a lot more thinking ahead of time to collect a sample. Some of the common DQOs typically include trying to estimate the mean, say, contaminant concentration or commodity concentration of something within a targeted decision unit area and volume of targeted media. So you hear me talk about the term, or use the term decision unit or DU often in this presentation. So in this case, the sample is prepared by collecting and combining groups or increments of individual elements into a single bulk sample for processing and testing. So you're only preparing one sample, you're collecting it from many points. Some of the variabilities you have to consider when you're doing this are the mass of the sample, total mass that you collect, the number of increments or points, testing points that you include in the sample. Even the collection method in the field is very important in this case. Things you don't normally have to think about when you're testing finite element media. So what I want to do next is let's look at the use of finite versus infinite element sampling methods for air, subslab vapors, soil, and sediment. We could do the same thing for looking at testing of surface water. And groundwater is a different story, same problems. We haven't really thought about that in much detail. I'm not going to talk about testing surface water today, just in focus on the time that we have. It's going to be very similar to testing for air. So let's look at different test methods for each one of these media and the implications. Let's start with testing of indoor air. Actually, the indoor air people have a very good handle on infinite element media sampling methods, even though they don't use the same terms. Let's start with the first, the risk question. It says concentration of X in the air of this room during the time period Y exceed a screening level or a risk Z. So that's where it's important to always start. First, start with develop your risk question. Something we don't actually do a lot in the environmental industry. We go out and collect a lot of samples, get the data, and then think about what kind of questions the data might be able to answer. But it's important to start with the question before you go out in the field and collect your samples. So first, thinking about data needs from the field standpoint, now you're telling someone to go collect samples. They need to think about what is the mean concentration of X in the indoor air over a specified, say, 24-hour period in this example. Well, in this case, the decision unit is the volume of air circulating within the structure in that targeted area during the specified time. This is our decision unit. To calculate the volume, we can look at the indoor air volume times the indoor air exchange rate times the designated time period. Just using this example, this is the default volume indoor air space for a single-family home, 244 cubic meters. So times, we'll assume an indoor air exchange rate of 0.35 times per hour. That's the minimum indoor air exchange rate recommended for residential structures. Times 24 hours, we come up with a decision unit or DU volume of 2,050,000 liters. So what we need now, from a data standpoint, we need to know the mean concentration of X. Maybe it's tetrachloroethylene or some other volatile chemical in indoor air for this targeted DU volume of 2,050,000 liters. So how do we do this? In this case, the indoor air people would never use finite element media sampling methods where they're collecting discrete samples within lots of parts of a room or something and running statistical tests on it. So the indoor air people are very far advanced in terms of environmental work. They go straight to what we would consider an infinite element media sampling method. Our target DU volume, 2,050,000 liters. Just to put this in perspective, just think of a beach ball that's as tall as a 5-story building. That's the volume of air that they need to get one concentration for that targeted chemical. Ideally, you would send the entire DU volume of air to the laboratory for testing, but no one's going to take a 16-meter tall suma canister in a laboratory, of course, not realistic. So in reality, you have to collect a representative sample over 24 hours of this targeted DU volume of air. Common way to do this is to put say a 6-liter suma canister inside of a building, as you see in the picture on the right. And you set it so that it draws a continuous sample over 24 hours. Now, this has some assumptions behind it. It assumes circulating air passing by the collection point is representative of the indoor air within the targeted area of the structure as a whole. Also, it assumes the targeted day where you collected the sample is representative of a long-term mean. So you can test these assumptions by collecting concurrent replicate samples from other parts of the room, maybe at different heights in the room or different parts of the house. And you can even test on different days to look at temporal variability. Either way, it's a good example of infinite element media sampling methods the indoor air people, again, have been using for decades. Let's look at testing subslab vapors. So in this case, say we're concerned about vapor intrusion into a building impacts to indoor air. In this case, the risk question might be, does the concentration of X in vapors intruding the building during time period Y exceed screening level or risk Z? So in this case, the subslab vapor screening level could be calculated by taking the indoor air screening level and dividing it by some attenuation factor. So what are our data needs for this then? From a field standpoint, the risk assessor, I would say, is telling me what I need to determine what is the mean concentration of X in intruding vapors over a 24-hour period. Or first I need to designate the decision unit. So in this case, the decision unit is the volume of vapors intruding the structure during the specified time period. We can actually calculate this volume by assuming a default vapor entry rate and multiplying it by the designated time period. Default vapor entry rate for vapor intrusion in cold climates anyway is 4.5 liters per minute. So we multiply this by 60 minutes per hour times 24 hours. So our decision unit volume is 6,480 liters per day. So from a sampling perspective, we need to collect a representative sample of this 6,480 liters of vapors that could intrude the building and impact indoor air. Let's look at the difference between using finite versus infinite element media sampling methods to collect subslab vapor samples. In this photo, the yellow image here is the approximate area where that would include 6,480 liters of subslab vapors. This is just a general approximation, assuming cylindrical draw area, which isn't realistic in most cases. Just an example. You'd say that the vapors are in the first half meter of subslab fill and soil, and assume a 20% airfill porosity. Just for examples. Ideally, then we would designate a hypothetical or even a suspect vapor entry point in the floor, drill a hole through the floor, and collect this entire 6,480 liters of vapors and send that to the laboratory for testing. And that would represent the vapors intruding that point on the floor during a one-day period. In reality, of course, we can't send that. That's, again, going to be a very big sample. So we need to collect a representative sample of those vapors from that point. So the question here is, traditionally what we've been doing, what I've certainly done a lot in my career, is go to this point, drill a hole through the floor, and then collect a single discrete sample in a 1 liter SUMA canister or maybe a 6 liter SUMA canister. But if you look at the comparison of the area for these types of small samples compared to the overall decision unit volume of vapors that we need to get a representative sample from, it's really minuscule. So these small volume discrete subslab vapor samples would only be representative of intruding vapors if the subslab vapor is relatively uniform. Well, in reality, subslab vapors or vapor plumes under slabs are never uniform. And that's a problem when we're using these finite element media sampling methods, or discrete samples, as we call them in the environmental business, to test for vapors under slabs. They have some use. Sometimes with discrete samples, we'll mention later, they're useful for identifying large-scale patterns. But at individual points, they really can't be relied on. So in reality, VOC concentrations in vapor plumes under slabs can vary by orders of magnitude over a very short distance. So these small volume discrete subslab vapor samples can't be considered reliable for calculation of an indoor air subslab attenuation factor. The subslab attenuation factor is going to vary depending on where the sample is taken, just because of the heterogeneity of concentrations in the VOC plume. So imagine if I have an indoor air data sample, which the indoor air people I can assume gave me some good data. I collect a sub-slab sample, 1 6-liter sample, at point X in the bottom right-hand corner, and look at the ratio of indoor air at VOC concentration in sub-slab to indoor air. Or I would get one attenuation factor. If I collect another sample at point X in the upper right-hand corner and look at compare the data between sub-slab vapor and indoor air, or indoor air sub-slab data, I'd get a completely different attenuation factor. So these attenuation factors we calculated are random and can't be considered to be representative of true conditions. A big problem with vapor intrusion is the vapor entry point generally isn't unknown. Even if you did know where the vapor entry point was, these small 1 to 6 liter discrete sub-slab vapor samples, they're too small to represent the intruding vapors. Remember, we're assuming thousands of liters of intruding vapors a day. So with the chance that a 1 to 6 liter discrete sample represents those vapors is fairly slim. So in the end, the error in estimation of a true sub-slab attenuation factor for a building isn't quantifiable. And California EPA, among a lot of other people, recognized this problem early on, that this attempt to use single sub-slab vapor samples compared to indoor air and calculate attenuation factors is only viable if the subsurface is reasonably homogeneous, which we know it's not. This has some important implications. It's in a paper we published in 2014, Estimation of Generic Sub-Slab Attenuation Factors for Vapor Intrusion Investigations. The first part of the paper looked at US EPA's empirical vapor intrusion database. In this case, in their database, they would collect an indoor air sample of hundreds of houses and then compare that to a single 1 to 6 liter vapor sample collected beneath the building slab. So as we just pointed out, the attenuation factor that they calculated for each building is completely random and unreliable. So the database itself isn't scientifically defensible for development of generic attenuation factors. The calculation of a 95% UCL based on this data is definitely what I would call fake data. So you can't fix this with statistical analysis. Imagine if I went to a football stadium or something and I wanted to estimate the average age of the people there, so I randomly picked 100 people, asked their age. I can calculate a mean and a standard deviation and 95% UCL and such, but then I find out later on that they all lied, or a lot of them lied. I have no idea how many. What can I do with the data? Well, you can't do anything with the data. Your database is no good. Statistics can't help you. So the US EPA's empirical vapor intrusion database, which I certainly spent a lot of time on early on, is what we'd call it's a useful but failed scientific study. There's a lot of comments I hear now where the vapor intrusion models don't work. See, the data don't match up. It's not a problem with the models. The models are very elementary physics, just basic physics. It's a problem with the sample data. We discussed this problem in the paper we published. We also discuss default vapor entry rates in different climate zones and indoor air exchange rates and attenuation factors for different climate zones based on work that building engineers have done looking at building energy efficiency and ventilation since the 1940s. They have a much better idea of building leakage, as they call it, the attenuation factors and such. So how do you collect a representative sample of sub-slab vapors then using an infinite element media approach? Well, this was worked out many years ago. It's a paper published by Todd McAlary and his colleagues in 2010. They called it high purge volume sampling, a new paradigm for sub-slab soil gas monitoring. In this case, a suspect or hypothetical vapor entry point is designated in the slab of a building. Typically, if you don't know anything about what's in the building, you might go to the center of the slab and assume that's where the highest concentrations are. There are a lot of variables in this you have to think about. Maybe you know where a source area is, another part of the slab. Then you designate a vapor entry point there to test. So then you would drill a hole through the slab just like we would normally do and collect a vapor sample, except in this case, you're going to purge the targeted decision unit volume of vapors from that point. Remember before we talked about, at least for cold climates, your decision unit volume of vapors, if you're interested in the average or mean concentration of a VOC and vapors intruding a building over a single day, is 6,480 liters. Well, that's going to be a big sample. Obviously, you can't send that to the laboratory. In this case, you would collect a continuous sample and assume a canister that's connected to the purge train. And now you would get a single sample from the laboratory, and the data represents the VOC concentration, or the mean VOC concentration, true VOC concentration, in vapors that would hypothetically intrude through that point over 24 hours. Now, typically when we do this work, we do 5 purges from the same point, because we want to look at vapor intrusion into a building at that point over 5 days, so that we can capture any variability in vapor entry into the building over a week-long period. Now, the exact source of the vapors in the building doesn't matter. This is something Todd and I talked about quite a bit. It's something he was interested in from a remediation standpoint. But we said, from a risk standpoint, the building doesn't care where the vapors are coming from under the slab. We just care what the concentration of VOCs are in that vapor. Another issue that we added to Todd's approach was this targeted DU volume. What volume of vapors do we need to purge from a risk standpoint and get data for? Otherwise, it's the same thing that he's always been doing. This is what this looks like in the field. This is a pilot study we did at a former dry cleaner site. The building's gone, and that we refer to this type of sampling as large volume purge, or LVP sampling. So in this case, it's very similar to a soil vapor extraction pilot test that environmental consultants have been doing for several decades. You drill a hole through the slab at the point, your hypothetical vapor entry point, and then you hook this purge line up to, in this case we used a shop vac, that's what Todd McKellar uses, and then you purge 6,480 liters of vapor from that point. So along this sampling train here, we have a SUMA canister connected to the purge stream. So the person would turn on the shop vac, it starts purging, and there he or she is monitoring the vapor volume with flow meters. We open the SUMA canister at the same time, and it's collecting a continuous sample over, say, the 10 minutes it takes to purge this 6,480 liters of vapors. That's the first day's sample. At the end of the first day's sample, you turn off the shop vac, close the SUMA canister, take it off, and hook up your next day's SUMA canister, or the next SUMA canister to it, kind of represent day 2 or DU volume 2, and repeat it. Collect a continuous sample during this second day, or this second DU volume. You do that 5 times. This gives you data for vapors that would intrude through that point over a 5-day period under the assumptions of vapor entry rates with building pressure differentials and such. Here's the results of the study we did. So again, the left-hand side, the picture of the slab here, the building's gone. We designate our LVP point for the center of the building, just as a default worst case scenario. We could have done it several points or put it at different points if we thought it was a source into one part of the building. And then we ran 5 purges, collected a single continuous 6-liter SUMA canister during each purge. The interesting part here is that intuitively, if you're used to the discrete sampling method we've been using in the environmental industry for years, you think, well, the more I purge, I'm going to start diluting out the vapors. The opposite is actually true. We see the same thing in soil. You look at the results here from our LVP for PCE. Sample number 1, 17,000 micrograms per cubic meter. Our screening level for commercial industrial buildings is 8,000. So that's obviously a problem. Sample 2, 36,000. Sample 3, 50,000. Sample 4, 51,000. Sample 5, 54,000 micrograms per cubic meter. Concentrations are going up in the samples we collect the more we purge. So this is really interesting. How can this be? The thing is that as we purge more and more, we're actually starting to capture outlier hotspots that we would have otherwise missed under the slab with collecting a few small volume discrete soil vapor samples. This actually, is another part of this study. We actually did a high density passive soil vapor sample study through this slab to try to identify the exact plume. And the initial discrete small volume sample, soil vapor samples collected on the slab completely missed where the plume was, in the high spot of the plume. I won't go into that in detail on this. We have a presentation and recording posted to our YouTube page. So it's something we very commonly see is these single LVP samples. The concentration of the VOC in the sample often exceeds the mean of the surrounding discrete soil sample data, or soil vapor sample data, because the LVP is capturing a larger volume and you're capturing these small hot spots. you'd otherwise would miss, and incorporating that into the mean. There's some references for that study. So published through a report just on our webpage, 2017. There's a recorded webinar with the presentation of the slides that we gave, and it's incorporated into our 2016 Technical Guidance Manual for Testing of Soil and Air and Water. We haven't published a paper on this yet. We're waiting to find a few more places to collect additional samples and get some new ideas. Doing this for petroleum is an issue because you don't want to use a shop vac to pump petroleum vapors out of the ground. As some people who've done this would find, you're going to blow up the shop vac if you get explosive levels of vapors. So you have to use an intrinsically safe pump. And so there's different sampling methods that can be done. We just haven't done it yet. So that's air, where they have a great concept of infinite element media sampling methods, and soil vapor, where we're getting there. Let's look at soil and sediment sampling next. This gets to be a real challenge, trying to collect a representative sample. So imagine this, you see in the bottom left-hand corner, here's a handful of soil, weighs about 1 kilogram. Just assuming these are all, on average, 1 millimeter particles, there are about 1 million particles of soil in that sample. Imagine handing this to the laboratory chemist and saying, tell me if there's 1 particle of PCBs in this soil, which would equal 1 milligram per kilogram, 1 ppm. Just imagine the difficulty in doing that. How are they going to even subsample that, collect to test? There are a lot of issues that come up. So you have to start basically back with the risk question. What do we want to know? This applies to any type of sampling, whether they're doing water sampling, fish sampling, soil. Start with thinking about what you want to know specifically. In this case, it's does the concentration of X in the area or volume of soil or sediment, Y, exceed the screening level of risk Z. I'm not going to talk a lot about sediments this time, but testing sediments has the same issues as testing soil. Just because you cover the sediment with water doesn't make it a different type of media and different type of sampling methods and uncertainty and such. It just may make it more challenging to collect samples. So here, you really have to start back with step 1. In step 1, you go through a thorough systematic planning process, as we discussed in section 3 of our technical guidance manual. Here's step 1 is to determine the contaminants of potential concern and your primary environmental concerns. Are we more concerned about direct exposure? Is it human health or ecological risk? Are we concerned about leaching concerns or leaching hazards or vapor intrusion? All this soil vapor data, much better to assess that, or other types of environmental concerns. Then you specify, you develop site investigation objectives. What are the specific questions that you're asking for this site? The next step or point is to designate your data resolution. I like to think of this as sort of satellite photos, your camera, your cell phone camera resolution. Here you're designating the decision unit that needed to answer the investigation objectives. And then you need step 4, the next step is you collect a representative sample from each area. So really the data resolution decision units is the first critical step. And these have to be based on answering your site investigation objectives, your DQOs. Do you need to test every spot from a risk assessment standpoint, or maybe every playground in a park, or maybe the entire park? Ideally, you get the site investigation, you develop the site investigation objectives to use in conjunction with discussions with risk assessors, remediation experts, etc., prior to the collection of samples in the field. The reality is that risk assessors and remediation experts typically get stuck with the data given to them with no time or money to collect additional data. Again, I would harass myself from this because on projects early on in my career, I was the one collecting the samples. And then I was the one reviewing the data. And I was never satisfied on either end that I was getting the right answer. So ultimately, it's all about this decision unit. We spent a lot of time discussing this in our technical guidance manual. We have a 6-part webinar series, training for doing what we call a decision unit and the multi-increment sampling methods. And one of the webinars is completely dedicated to talking about decision units. How do you split a site up into targeted areas for collection of representative samples? A good way to think about a decision unit, or it's an area and volume of soil you would send to a laboratory as a single sample for testing if you could. So decision units should always be designated for testing, regardless of how you're collecting the sample methods, regardless of the media, whether it's finite or infinite element media. This is always your first step. And we've actually been doing this in the environmental industry from the beginning. We just haven't assigned a name to it or used it as part of designing the sampling scheme, at least using it properly. So we talk about spill area decision units in the environmental industry for contaminated land where we suspect high concentrations of contamination. From a risk standpoint, we talk about exposure area decision units. It could be a playground or your front yard or an open area of a commercial building where people are exposed on a routine basis. We talk about boundary or perimeter DUs, where we're trying to identify the lateral or vertical extent of contamination around suspect contaminated areas. These are all types of decision units. We want to go into each one of these decision areas now and collect representative samples for those targeted areas and use the data to make decisions. Let's look at the difference between attempting to use finite element media sampling methods versus infinite element media sampling methods to collect representative samples of soil and sediment. And here I worked with Dina Crumling, a recently retired geologist with US EPA headquarters, Superfund Group. And she tracked down a lot of the old guidance documents published by EPA in the 1980s and 1990s, where they were initially developing the sampling methods for soil. This is a document from 1985, Field Manual for Grid Sampling of PCB Spill Sites to Verify Cleanup. As far as we know, this is the first document where the idea of Discrete, which is the finite element soil sampling methods, were developed and published by EPA for use at contaminated sites. One thing to point out is no other industry, no other sampling intensive industry like the mining industry, agriculture industry that tests soils or crushed rocks and such uses discrete samples. They're only used in the environmental industry. The reason was, as we discussed in the paper we published, is the people involved in preparing this guidance at the time didn't have any training or really background in infinite element media sampling methods. We'll discuss a little more about that later. Also at this time, risk assessment guidance was being published. The idea of risk-based screening levels was coming out. And the early sampling guidance, the documents, people writing this guidance were confused about the applicability of screening levels to any testable mass of soil versus the mean targeted exposure area. So that was one of the issues why they started testing individual points within targeted areas because they weren't sure about how the screening levels apply. We discussed this in the paper we published in 2017. I'll show you more details later, a critical review of discrete soil sample reliability. So some of the key assumptions that were in these early documents for the reliability of discrete soil and sediment data include in US EPA 1985 document mentioned is the level of a contaminant in soil is assumed to be uniform within a contaminated area and zero outside of it. So if this was true, the concentration of a contaminant anywhere within some spill area should be relatively uniform. This made sense, I think, from the people writing this guidance. They were familiar with industrial wastewater, liquid spills. So if the concentration is uniform in the liquid, it should be relatively uniform in the soil. 1989 document published by EPA, when there is little distance between points, it is expected that there will be little variability between points, probably for the same reason. So if this was true, then a few samples collected from a few single points within a targeted property should be adequate to identify the extent of contamination and assess risk. So the data for individual points could even be compared to risk-based screening levels, since in theory they represent a larger area, might even be an exposure area. Now, one interesting point is they never tested this in the field. And if you read the original documents, including the ones I'm pointing out here, you can tell the authors wanted to go out and test in the field. Is it really uniform? Let's collect lots of individual co-located samples and see how uniform it is. They never did this because they didn't have the time and I guess the funding. And that's why in their document they very clearly state that this whole sampling approach we're discussing may change in the future as new information is gathered. It took 30 years, but that's what's happening. So do you ever wonder, I certainly did this back on my geologist side, what if I move my sample point over a few feet? Would I get the same number? How uniform is it? How close would it be? What if the laboratory tested a different subsample of soil from this 100-gram 4-ounce jar of soil I sent? This idea that laboratories homogenize your sample. As a chemist, the laboratory told me, try sticking a metal rod in a 4-ounce jar and stirring it, and let me know what you think. Basically, you can't homogenize soil. And even in theory, what happens more often than not is the fine particles settle down to the bottom of the jar. And when they collect their small mass, 10 to 30 grams for stuff like PCBs, as little as 1 gram for metals, from the top of the jar, then you're missing higher contamination that's in the fine material that's actually posing a greater risk. It's always been an issue. I don't know any laboratory chemists that would disagree with that, but that's what they're told to do. So we carried out a field study of discrete soil sample variability. We picked 3 sites in Hawaii, and then using grant money from US EPA Region 9, we collected hundreds of co-located discrete samples at each study site. So at each one of these sites, we designated 24 grid points. Around each grid point, within about a 1 meter square area, we collected 5 co-located discrete samples. We had these processed at the laboratory using multi-increment sampling methods, where they would dry it, sieve the sample, and then very carefully subsample it, collect a minimum 10-gram mass from 30 points, and test that. So we're confident the data we get for each discrete sample is reliable. The data the laboratory has given us is representing the sample we sent them. We also took one of the samples, had it tested 10 times by the laboratory. So the first case, we're looking at what we call inter-sample variability between co-located samples. In the second case, we're looking at variability within a single sample to see how far off the lab data could be for the sample we submitted. I want to discuss it in this presentation. We also collected what we call multi-increment samples at each one of the sites. The multi-increment sample, you take your target area, you collect 1 sample for a minimum of 50 points. The sample has to weigh minimum 1 to 2 kilograms. We'll discuss why later. And then the lab just tests that 1 sample and they process it carefully. We'll actually collect 3 independent multi-increment samples in the same area, compare the results to look at variability. 3 different sites we looked at. The first was contaminated with arsenic from wastewater from a wood treatment facility. It was dumped onto fine-grained soil, study site A. Here we expected variability to be relatively low compared to the other sites. Site B was incinerator ash contaminated with lead mixed in with fill material that was spread out around this incinerator to fill in low areas. We expected variability between co-located samples at this site to be higher than the one with arsenic, contaminated by arsenic and wastewater. Study site C was a site contaminated PCB from transformer oil, electrical equipment oil. It was dumped onto the ground. We already knew from collecting this site and other PCB sites, this would probably be where we see the highest variability between different sampling points or co-located samples, and even within single samples. These field study results published in a paper in 2017, a critical review of discrete soil sample reliability. Part 1, field study results. Part 2, implications. Interesting thing with the co-authors here, John Purge, it's the one that raised the issue of sampling when he joined our office in 2004, early 2000s. He actually has a farming background kind of as a hobby in addition to being an environmental scientist. He knew from a farming standpoint you collect 2 samples of soil side by side and test for things in the ppm level that can be different. Marvin Heskett's a laboratory chemist, ran the large laboratory here. He knew from his work that you could test different subsamples of soil in the same sample submitted to a laboratory and get completely different results. They're a great team to do this. We have a webinar, posted several webinars actually on our webpage on the field study. We'll go straight to the results. So again, in each one of these sites, we tested co-located samples around 24 points. For study site A, contaminated with arsenic, with an average total variability within the sample between co-located points about 2-fold. This is real data. You see one sample, 120 ppm, the upper right-hand corner, total arsenic in soil, but just 1 or 2 feet away from it, sample is 260 ppm arsenic in soil. This variability is completely random. If you went to the other side of the 260 ppm sample and collected one, it might be 100 ppm or something. There's no trends in this. Another issue is we saw the same variability regardless of the concentrations, even at ppb level, ppm level, very similar variability within the samples, within co-located samples. As we predicted at the lead site, the variability increased between co-located samples. The average total variability 7-fold. So you see example here, 120 ppm, coincidentally in the upper right-hand corner again, But then just 1 or 2 feet away, concentration of lead in discrete sample 800 ppm. So quite a bit of variability. And again, it's completely random. Again, you see similar variability, low concentration, high concentration. PCB site, as we predicted, the total variability was much higher. Average was just 39-fold. And again, this is just the minimum variability of discrete samples between co-located samples from single points. So in this case, one discrete sample, 4.9 ppm PCBs, and then just 1 or 2 feet away, 91 ppm PCBs. So again, very random variability. It's not indicating a trend. The sample we tested for split up into 10 subsamples, tested each subsample for PCBs at this site. The mean of that was 2,400 ppm PCBs. So this is completely random data. That's a true hotspot. It's only about a few inches apart as far as we can tell. One interesting thing back to the lead side is in Hawaii we use a screening level for lead in soil residential use of 200 ppm. Out of the 24 points we designated for collecting co-located samples, at every single point we could have collected a discrete sample and gotten a concentration below 200 ppm or above 200 ppm. So someone could hypothetically go into the site, collect a set of 24 samples, and they're all below our screening level. Another person come in, collect a sample, or collect 24 samples from the site, and all 24 samples are above our screening level. So why is this happening with this high variability we see in environmental data, in this case for soil? For soil, the first issue to think about is referred to in sampling theory as compositional heterogeneity. This is heterogeneity between particles in the sample or in the soil itself. So in this case, these are photomicrographs of the soil at the PCB site that we were testing. We were wondering, why are we seeing such big variability in the sample data? What turned out to be the case is there are these small nuggets of PCB-infused tar in the soil. So if you look on the bottom right-hand corner, here's the soil sample. We saw these bits of soil in the sample that were darker and easily crushed. Didn't look like the rest of the soil. We zoomed in on them, and they're shaped like a tic-tac candy. Or as we say, if you're familiar with tropical environments, they look like a tiny roach egg, 1 millimeter or so across. So we zoom in on these small particles in the upper left-hand corner. What they are, we're quite sure, they're dried up beads of tar, PCB-infused tar that migrated into the soil. So if you take we show this in the workshops we give on this study, take some olive oil and dump it onto a plate of dry flour and watch what happens. The oil will beat up and then it'll sink into the flour. It's exactly what happened here. So of course it often comes up when we're testing for contaminated soil is what's the maximum concentration of contaminant in the soil? Well, think about it. In the upper left-hand corner here, if I could go in with an electron microprobe that could test for PCBs or something, if I tested a small enough spot, then I'd test an individual particle of PCBs. So if the contaminant is present in the soil, the maximum concentration at some scale is always 100%. It's always 1 million parts per million. If it's not in the soil, it's 0. So there's an easy answer to the conundrum I used to always wonder about from risk standpoint, maximum concentration. It's easy. It's either 0 or 100%. Anything in between, you're just diluting it with soil. Now, from a risk assessment standpoint, the objective is always to determine the mean concentration or the true concentration of the contaminant in the designated DU area and volume mass of soil as a whole. So an important takeaway from this slide is that concentration varies with the mass and volume of soil tested. If the laboratory tested 1 gram subsamples of PCB contaminated soil, they would get one range of concentrations, very high. If they tested 30 grams, they get another range of concentrations. The highest would be lower because they're diluting out these nuggets more. The second factor that is one of the main causes between this high variability we see between co-located discrete samples is referred to as distributional heterogeneity. This is just a variability in the distribution of contaminants within your targeted area. I like to use this photo of Jackson Pollock. He's famous for a splatter painting on the left. Zoom in and look at his painting. That red dot is actually the size of a typical discrete sample, a 4-ounce jar, 100 grams. Imagine moving that red dot around just a few inches or a few feet. You get completely different concentrations of black material. So you can start imagining how random the concentration might be for any given individual discrete sample. On the right is a milk truck that flipped over and the milk ran down the embankment. So imagine this if you couldn't see the milk. And think about this especially from subsurface testing. And someone during the presentation brought up problems with testing for VOCs in the subsurface and why are the data always so far off? Why do you miss so much contamination? Think of this picture here, flip it on its side as in a cross section. What's the chance that a few discrete soil samples where they're testing 5 grams of soil at the laboratory would capture the contamination in the subsurface? In most cases, you're going to miss the main contamination. You're going to underestimate the mass of contamination present as well as the distribution. This compositional heterogeneity, distributional heterogeneity, among other issues, are the reason or primary cause for failed confirmation samples, the discovery of new contamination after you think you've cleaned up a site. So the bottom line here is that the laboratory data for a sample you submit are not reliably representative of the sample that you submitted. I think any laboratory chemist would agree to that. And also the sample you submitted that you collected in the field isn't reliably representative of the area where it was collected. So at a minimum, we would say twofold variability. It could be way, way worse than that, even several orders of magnitude different from that immediately surrounding area. So this problem can't be fixed by collecting more and more discrete samples. What you're using, when I say you, I'm talking to myself, my past anyway, geologist, is using the wrong sampling method, for this type of media. The problem can't be fixed either by statistical evaluation of discrete sample data. It can help, and that's all the risk assessors could do because they're getting this data dumped on them in the first place. But you can run into a lot of problems with that. One key flag that you see at sites where a lot of discrete samples are collected are these isolated hotspots. We've always wondered about how do we magically find these hotspots with one sample. As we discussed in the webinars and the paper we wrote, these hotspots in most cases are complete artifacts of the sampling data. If you went back to the same spot and collected a sample a few inches away, you might not find anything. So it's just an artifact of the way the samples were collected. And in most cases, not reliable, even though we've been using it for cleanup for years. There are a lot of early warnings about this discrete sampling method, even in the late 80s and in the early 90s. Here's a quote from an EPA guidance written in 1992. The so-called grab sample was not really a sample, but a specimen of the material that may or may not be representative of the sampling unit, or what we call now the decision unit. Great care must be exercised when interpreting the meaning of these samples. The problem back then was they didn't really have a solution to it. So what was the solution? You just collect more and more samples until you run out of money, and then you do what you can with the data you have. Which, in a lot of cases for soil, I would say probably we're capturing the worst contamination out there, where any spoonful would fail. But we probably missed a lot of contamination where you have high heterogeneity. But the risk assessors almost got it right from the start. They understood high variability of contaminants in soil. And from a risk standpoint, what we're concerned about is the true concentration of contaminant in some specified area or volume of soil, or the mean concentration. So in this example here is a front yard, 20 feet by 20 feet, 2-inch depths of soil. So really you have to start thinking in terms not just area but volume when you're out testing in the field. Here the volume is pretty small. It fit in the back of a pickup truck, 2.5 cubic yards, about 2,500 kilograms. So that's our target. Ideally, again, thinking back to what we talked about for air and for soil vapor, ideally we would excavate and send the entire volume of exposure area soil to the laboratory for testing as a single sample. They would give us a single concentration, say of lead or PCBs or whatever, and we'd use that data to assess risk. This isn't practical in most cases, of course. Although we have done this when we're looking at testing sediment in storm drains and such. We just collect all the sediment in the storm drains, send it to the laboratory, and they'll process it properly and test it. Here's an alternative. This was an alternative forced on risk assessors by people like myself, forcing it on myself, where we would collect discrete samples thinking that we were trying to determine the extent of contamination with discrete samples, which we now know isn't reliable. But here's what the risk assessors got. They get ideally at least 10 discrete samples, and the data has already been given to them. The laboratory has independently tested each sample. So what the risk assessors do, the only option they have is use statistical analysis to address the variability, estimate the mean. This isn't a sampling approach that risk assessors came up with. It was something that was forced on them, I would say, back in the 1990s. And it's just the way things have always been until recently. So the problems with this, a lot of times it cites 10 or more samples from a targeted area aren't available. Risk assessors never see this. This is probably 99% of the sites, though. In this case, people in the field would just use the maximum concentration. We already saw how that is really meaningless in terms of the massive soil tested and reliability to begin with. It doesn't mean anything in terms of risk anyway. Also, laboratory analytical costs often limits the number of samples collected. It's a true joke in the field. How many samples should I collect? Where you take your budget allotted for laboratory analysis, divide that by the cost of analysis, and there's the number of samples you collect. That's why we did things my first 10 years of my career. So again, there's no science behind that. It's just the way things were. Important point, especially for field people, is that understand statistical analysis of data only evaluates the precision of the statistical test employed to estimate a mean for the sample data set provided. The representativeness of this single set of data is unknown. If another person went back to the site, collected another set of 10 discrete samples, would they come up with the same mean, the same 95% UCL? We'll look at that in a minute. So this gives you a false sense of data precision. The statistical tests do if you don't really understand statistics in general, even when using a 95% UCL. Let's look at that. Each one of our study sites, again, we had hundreds of discrete samples from 24 points at each site. What we did is we put together random sets of 10 samples each for each study site. We did this 20 times for each of the study sites, so 20 sets of replicate discrete sample data. Calculated a 95% UCL using ProUCL for each one of these 10-point sets and compared those. Now, if contaminant concentrations were really uniform, like they assumed back in the 1980s, the 95% UCL should be very similar. At the arsenic site, the variability we see in 95% UCLs from 10 sample sets range from 403 ppm to 776 ppm, almost twofold variability. Again, this would never take into account in the risk assessments. Site B, similar, a little bit more than 2 times variability in the 95% UCLs for independent sets of discrete samples collected there. Site C, the PCBs, which are what a difficult type of contaminant test in soil. We still have difficulty with it even when using our multi-increment sampling approaches, collecting huge samples. 95% UCLs, independent sets of discrete sample data range from 9.4 ppm to over 1 million ppm. So think about that in terms of your real precision and certainty in data. So again, this is a big issue. This type of field sampling area is never directly considered in traditional risk assessments because no one ever collects replicate sets of discrete sample data to verify that the data collected, or samples collected, are actually representative of field conditions. If you look at the EPA's guidance on data validation and verification, validation only deals with the laboratory error, which the laboratories have good control on that. The verification part is intended to apply to the representatives of samples collected in the field, but there's no discussion of potential error like this. The actual document which we discussed only says we'll make sure they follow the sampling plan. So there's never in this type of industry testing soils, there's never been an approach to verify that your data are representative in the first place. Here's a much better approach. Here's what risk assessors would do. If we got together in the 1990s when I first started and thought about it more, I don't know why it took me 10 years to think about it and meet people who did. Here's the way to assess, to sample contaminated soil using infinite element media sampling method in terms of risk. So we'll go out, we're going to do the same thing. We're going to collect a little bit of soil from lots of points within the area. But our next job is to determine the mean concentration of a contaminant in that soil that we collected. We just want one concentration. Well, the best way to do this is to combine all the soil together in one big sample, send it to the laboratory, have them subsample it and process it very carefully, and give us one concentration for that soil collected. The idea of testing individual points of soil that we collected is purely from the field people trying to estimate extent of contamination and such. We would never do that from a risk standpoint. It'd be totally illogical. So this gets to the basics of sampling theory for infinite element media sampling methods and what we refer to as multi-increment samples. So again, we're just going to, in this case, the estimated mean concentration for the DU is directly reported by this one sample. We're going to test our precision, reproducibility of our data for the soil. We'll collect one sample from a lot of points, and then we're going to go back and do it again, and then we're going to go do it again from independent points. Now we have 3 samples from our exposure area. We compare the data for each one of those samples, get an idea of what the total precision of our sampling and test methods are, and get much more confidence in the data, or not confidence. The only questions that come up here then is how many increments should be included in a single sample, and also how should they be collected in the field. It's very important to collect core-shaped increments through your targeted depth of contamination or its soil. And also a big part of this is what mass of soil should be collected. This is the foundations of a Purge sampling theory for infinite element media. Again, they were working with mining companies, which we'll talk about in a minute, estimating commodity concentrations in ore. So this is the separate type of sampling method. It's a totally separate set of statistical methods. It replaces finite element statistical methods and sampling methods. So some of the considerations you have to take into account when you're testing soil and sediment and crushed ore and such, trying to collect a representative sample is, again, what's the minimum mass I need is called fundamental error to deal with compositional heterogeneity. I've got these nuggets of PCB-infused tar balls or nuggets of oil or lead in my sample. I get to the laboratory. Laboratory needs to test minimum mass of that sample just to get a representative number of those particles with high concentrations in them. Also, we have to think more about what the number of increments to collect is referred to again as distributional heterogeneity within the site. How we collect the samples, the increments, again, core-shaped plugs or soil in different areas over a grid type pattern. You also have to think about how the sample is processed when it gets to the laboratory. The laboratory doesn't open your bag or whatever and take out a random gram and test it. They have to air dry it, spread it out, and very carefully subsample it in the same manner that we did in the field. And that's what they test. And then there's some error associated with the analysis. That's the least error that you have to deal with. Most of the error in environmental work or any type of work involves how the samples were collected, not how they were tested, or at least the mass tested. So there's a lot of discussion behind this, but the bottom line is for testing soil and sediment, you need to collect a minimum mass of 1 to 2 kilograms of soil or sediment. This is based on a review of lots of databases, especially by the mining industry and others over the last 50 years. You need to collect a sample from at least 50 points. Again, it's based on a lot of field studies testing precision, or so 50 increments. And in theory, this allows part per billion level resolution, although some of the sampling theorists I talked to question that, that kind of accuracy, or at least ppm data resolution for your samples. A lot of details in this we're not going to go over, but it's included in our webinars and some of the books written on testing for infinite element media. Why did miners figure this out? Why'd they figure it out 50 years ago and the environmental people were just now trying to figure it out? The key thing in mining is they have true data verification and validation. So imagine you're a company that looks for gold ore. So you find a place, you dig up a bunch of rocks that you think has gold in it, you grind it up and you collect a sample, and your objective is you have to estimate the mean concentration of this commodity, gold or whatever, in your ground up ore. But ultimately you want to estimate the mass. What's the true mass of gold in this ore? Then you sell this crushed ore to another company that's actually going to extract the commodity, extract the gold. They're going to know exactly what the mass of gold or what that commodity was in the soil. So it's a great way to validate and verify your sampling data. Well, they knew since back in the 1800s, the initial discrete type sampling methods they were using for crushed ore could be totally wrong. And over the next 100 years, really, they progressively isolated different sources of error that we talked about before. Mass, shape of the particles, size of the particles, density, all these details. And that's where they developed the theory of sampling, first published by Purge in the 1950s. One issue is why this didn't get incorporated into environmental work is it was in French. They were doing most of the international mining until the 1990s, and very few people in US EPA and agencies apparently could read French or knew about this, although a few could, and they bring it up in the documents they wrote. Just urging some caution. So from a mining standpoint, the results of poor sample collection methods, if you underestimate the mean concentration in your mountain or pile of gold ore, then you're giving away your commodity. If you overestimate the mean concentration, well, you're cheating your customer. Either way, your customer is going to find out. Most commonly what's seen in the mining industry, and it's the same thing we see in the testing soils, poor sampling methods most commonly underestimate the true mean of the contaminant, the commodity, whatever you're investigating. The reason is, just like we showed in the soil vapor sampling, you're missing these small high concentration areas within your targeted area and volume of soil. Either way, in the mining industry, you go out of business. In the risk assessment environment industry, you don't, because it's much more difficult to determine or to identify poor sample data than it is, say, in the mining industry. In the agricultural industry, poor sampling data, your crops die, or you waste money on fertilizer and such. So think about the implications of this for risk assessment. I'm not going to get into the details at all. There's different classes on sampling theory. Chuck Ramsey is probably the foremost sampler of anything in the world that I know of. He's trained all the regulators and consultants here in Hawaii. He teaches a 4-day class. The first 3 days are kind of de-brainwashing us of using finite element sampling methods and discrete soil sampling methods for things like soil and sediment. He also teaches classes on food, soil or water. Francis Pittard is still the godfather of sampling theory in general, especially with the mining industries. He was teaching, I think, a 1 or 2 week long class on sampling theory. The best way is same way we did. We took the initial classes, but then our office actually has a field group. We go out and do our own field work. Start practicing in the field. We made plenty of mistakes over the last 15 years. Learned how to overcome them, and they're in our guidance. Here's our technical guidance manual published for this state. I like to refer to this as risk-based site characterization. We don't use the term in the manual yet, but that's really what we're doing. Different sections, section 3, 4, and 5 look at how to plan a site investigation. Section 4 is sampling theory, how to collect multi-document samples and process them, and the use, which we would use sometimes for discrete samples, and misuse. And then field implementation, how do you do this in the field? A lot of details, sampling tools, clearing sites, again, understanding the site history and such. We have a 6-part training series posted to YouTube. You can find it on our webpage. It goes step by step through each one of these sections and parts in the field, even including laboratory processing. So then with this, just from a good science standpoint, we challenge ourselves with trying to come up with reasons why, well, this won't work. And we already know initially what you'll think about why this won't work for soil is, well, we want to collect discrete samples so we know the extent of contamination at a site. Well, we've already demonstrated how you can be totally off on that. It can help to pick large-scale patterns, but your small-scale patterns based on single samples can be totally wrong. So you're much better off doing this decision at multi-document sampling method. So the other question that comes up is, what about acute risk? Back to that question in the 1980s, early 1990s, where the reason to collect discrete samples. Let's think about this for this site. Again, what's our risk question? Does the concentration of X, let's say lead, in any potential discrete soil sample collected from the yard exceed a concentration of Y? So hypothetical acute toxicity. A kid goes out, eats soil from that site, that particular point, and gets sick immediately or within a few days. Some of the sampling considerations, again, what's the mass of soil in this area? It's pretty small, 20 feet by 20 feet, but about 2,500 kilograms of soil. Think about that, a discrete sample is about 100 grams. Laboratory aliquot for metals is only 1 gram. That's a pinch. So we have our risk question. The next question is, what's our decision unit? So from a discrete sampling standpoint, 100-gram samples are 25,000 potential 100-gram discrete samples in this front yard. How many of those do you need to test to have any confidence that no discrete sample in that front yard would exceed your acute screening level. We don't really care about discrete samples anyway from a risk standpoint, or even from a laboratory standpoint. They're only testing 1 gram, not 100. It's a random 1 gram pinch of soil from the sample you sent. Now we have 2,500,000 potential samples in that front yard. How many of those do you need to test to get some degree of confidence? But really, from a risk standpoint, we're more concerned about kids eating the soil. The default soil ingestion rate, 200 milligrams. Now we have 12,500,000 potential samples or decision units in that front yard. How many of those do you need to test? A lot, I would say. Let's say we're just concerned about pica kids eating dirt. So 10 gram samples of decision units, and now we still have 250,000 potential decision units to collect and test, samples to test. So in reality here, it's an unanswerable question. It's a very reasonable question from a risk standpoint, acute health risk, but it's unanswerable in terms of me going to the field and collecting a sample or enough sample data in terms of feasibility and cost to give you any level of confidence. So testing for acute risk with a reasonable degree of certainty just isn't practical. Also, it's something to keep in mind, risk assessors know, most field people don't, is that there aren't acute toxicity factors for most chemicals. There are for some volatile chemicals, but for other chemicals in soil, there really aren't. So there's really no such thing as an acute screening level for most contaminants in soil. Even if there was, there's no way to reasonably clear even a small area from potential acute toxicity concerns. So if you're really concerned about it, say of bits of lead shot, like shown in the picture here, that have 40 milligrams of lead, what would that do to a kid's blood lead level if they swallowed it and it completely dissolved? Think about that. If you think you have this problem in your yard or in this exposure area, bits of paint chips or PCB infused tar balls, really all you can do is remove or cap the soil. So are discrete sample data ever useful? Well, yeah, we actually include guidance on using discrete sample data in our technical guidance manual. In this case, it was a 9-acre site contaminated with arsenic, again from water-based arsenic herbicides. We knew the site was heavily contaminated arsenic. In some areas, we actually did some DU-MIS sampling. It's going to take us forever to figure out the extent. So another consultant went out with the field XRF and collected 90 discrete samples from a grid across the site, tested each one, pretty quickly mapped out in a single day where the main part of contamination was. So we have very high confidence in this case. Liquid-based contamination, fine-grained soils, this area A you see in the left-hand picture is all heavily contaminated. Don't even bother resampling it. It's a risk problem. Area C looks like all the samples came up clean. We're pretty confident that area is going to pass the risk assessment. Area B is where you get into trouble. Here you see these isolated hot spots, again, would pop up within that area. If you retest area B, you'd see a similar number of hot spots, but they'd probably be in a different place. So in this case, what they would do when they remediate the site, the plan is just to remove all the soil in area A and B down to I think it's about 6 inches to a foot contamination, and then retest the entire site using DU-MIS methods, designated decision units. In this case, there were 1-acre decision units or smaller ones where the area of contamination was. Collecting MI samples just to demonstrate that the site is clean. Are discrete sample data ever useful? Part 2, no. This is a site 9 acres contaminated with PCBs. First red flag goes up here. You see all the isolated hot spots, and you'd see isolated cold spots too if we looked at that, based on 1 or 2 sample data points. And these ISO concentration maps, computer programs will generate these really pretty maps. In this case, they're completely artificial, at least in terms of these individual hot spots. At this site, they elected to go ahead and excavate these hot spots anyway, but then retest the entire area for PCBs just to confirm cleanup. And we cautioned them against this, to be honest, in the first place, because of our experience with PCBs. They excavated all the soil, pretty high cost, retested the entire area, and you see the DUs here. They're based on exposure areas, their contamination patterns and such. And the site was completely contaminated with PCBs still, even after digging out all this soil. The concentrations of PCBs in the individual DU areas, not surprisingly, were higher in general than the average concentration estimated by the discrete samples in that area. So this is how discrete sample data can really fool you. A lot of times you would never whoever redoes this. It's one of our requirements. People ask, well, how do we get this started? We let people use discrete sample data to clean up a site, but then we require multi-increment sample confirmation. We did this for the first 2 years. After about 2 years, consultants and RPs in Hawaii pretty much quit using discrete sample data, except for very rough screening in some cases. So I end up with a discussion of ITRC's incremental sampling methodology guidance. We were involved in preparation of this guidance in 2012. And by then we had 8 years of experience doing what we call DUMIS site investigations, and we helped with some of the initial writing. One problem is we didn't have a good set of field data, and this collecting MI samples and DUs is pretty obvious. The biggest problem we have is not understanding just how far off discrete sample data can be. So the problem with this initial guidance from ITRC is discrete sampling data, or MI data, ISM data as they call it, comes off. It's an option if it's available at your site. That's not the case at all. Actually, what we demonstrated and what's predicted by sampling theory very quickly is you can be completely wrong with your discrete sample data. If nothing else, you waste a lot of time and a lot of money. This is why it takes years and years to clean up sites in the US versus countries that went straight to DUMIS sampling data. So that's one of the problems with the guidance. We didn't have a lot of experience in. Most of the people on the team had no training in sampling theory, no experience in ISM methods in the field. You won't see any discussion of infinite element media sampling or finite element media sampling in this document. It lacks a critical review of discrete sample data. There's a lot of concern there about making discrete sample data look unreliable when we didn't have the data, field data to back it up. Well, now we do. It's also incomplete discussion of sampling theory, like I mentioned. It's a hypothetical evaluation of ISM data accuracy that's very misleading and inaccurate. It was interesting to do it again at the time, but it's quite wrong. It was an attempt to use finite element statistical methods to assess the reliability of G sampling theory for infinite element media, which is, again, it's completely wrong. That's why they had to come up with a totally different method of statistics and sampling methods for infinite element media, because you can't do that. It's currently being updated. I think it'll be improved. It'll still be limited. There's not a lot of experience with the people writing it, but they're very motivated. Actually, some of the people have a lot of experience now. So that was the end of the presentation. So I end it here. We had some questions come up. Really interesting. It's nice to get away from sampling dirt and even vapors. The first questions that came up were on testing fish. So, and even our office, we want to test fish for PFAS. Well, is this a finite element media sampling question or an infinite element media sampling question? Well, it depends on your question. If you want to know the variability of PFAS in the flesh of tuna with an individual fish, then it's a finite element testing problem as far as the overall problem is. If you want to know what the mean concentration of PFAS is in tuna coming into Honolulu Harbor fishing market every day, then it's more of an infinite element media testing problem, where in that case we would go out to the fish market. I'd designate some area of tuna fish and collect a small plug of tuna of flesh from each one of the fish, combined in a single sample, and have it tested for PFAS. I might do that 3 times on 3 independent sets of fish or something to test my sampling precision. All it depends on your question. That's where you have to start in the beginning. What's my question? What's my decision? What sampling method is most appropriate? Also, we're talking about testing for microplastics in surface water, which is really interesting, something we haven't thought about, but I'd love to think about that more. Anyway, thanks again for joining me, the people that did, and look forward to working on this problem again in the future. Here's one last note I'd like to add. This relates in a way to testing a fish we were just talking about. Let's think about testing bags of potting soils in the bottom left-hand corner of this slide. Let's say the company that made the bags of soil, and you wanted to ensure the nutrient content, so the organic carbon or the nutrients in each bag of soil, was up to quality standards that you were advertising. So how would you do this? This would actually involve both finite element and infinite element media sampling approaches. So first you would, from a finite element media sampling approach, you would pick a random number of bags, and there's different ways to do this, of soil to test. And then to test the soil in each bag, however, how would you do that? Well, you wouldn't just grab a random pinch or a random spoonful of soil out of each bag. Out of the bags you tested, you'd use an infinite element media sampling approach. So you'd take this bag of soil, each one you tested. Individually, you would break open the bag, spread the soil out, collect a subsample from that soil and then test that subsample for the soil. And then when you do this type of sampling, you need to meet all the requirements for multi-increment sampling and processing and testing and such, including minimum testing of 10 grams of soil. So this is just an example of where you'd be using a finite element and mixed infinite element media testing approaches. So you do this for each of the bags that you selected. You get a concentration or whatever it is you're looking for in each bag, and then you look at the variance of standard deviation. Maybe look at 95% upper confidence levels or other things, more from a QA QC check. So think about this when you're testing things. You can actually use a combination of both of these test methods or sampling theory methods, depending on the situation you're dealing with. Thanks.